The Src protein kinase acts as a molecular signal integration device. Src consists of two clearly demarcated regulatory domains, an SH3 domain and an SH2 domain. The catalytic domain is structured much like other tyrosine kinases. In addition, a linker and the short C-terminal tail play regulatory roles. To be an active protein kinase, the active site of the Src kinase domain must first become accessible to substrate proteins. In the inactive state, the active site is blocked by an activation loop. The activation loop must be phosphorylated, most likely through the actions of another Src kinase molecule, not shown here. Phosphorylation causes the activation loop to swing out of the way and rearrange so that other substrate proteins can bind and become phosphorylated. Src is kept inactive by an interaction of its SH2 domain with the C-terminal tail peptide. A phosphorylated tyrosine on the C-terminal tail is buried in a deep binding pocket in the SH2 domain. To activate Src, this phosphate group is removed by specific phosphatases. Upon dephosphorylation of the tyrosine, the C-terminal tail is released from the SH2 domain. In this cartoon view, Src is represented in its inactive state. The linker segment, shown in red, connects the SH2 and SH3 domains on the one hand and the kinase domain on the other. The activation loop, shown in dark green, drapes across the catalytic site, and in this configuration blocks the catalytic domain's access to the substrate of the kinase. Several other intramolecular interactions hold the Src kinase in an inactive configuration. The SH2 domain, in blue, binds a phosphotyrosine at residue position 527. The SH3 domain, in light green, binds the linker segment. When a PDGF receptor becomes activated by ligand binding, its C-terminal tail becomes phosphorylated on tyrosine residues. One of these phosphotyrosines can be recognized and bound by the SH2 domain of Src. The SH2 domain, which until now participated in an intramolecular binding, switches and forms an intermolecular bridge by binding the phosphotyrosine on the C-terminal tail of the PDGF receptor. The SH3 domain follows suit. It too breaks its intramolecular binding and binds to a proline-rich segment on the PDGF receptor. These shifts liberate the kinase domain of the Src protein. In a series of concerted reactions, the tyrosine residue at position 527 of Src is dephosphorylated, and the activation loop becomes phosphorylated on a tyrosine residue. This last alteration forces the activation loop to move out of the way so that it no longer obstructs the catalytic cleft of the Src enzyme. This allows Src to phosphorylate a diverse array of protein substrates on their tyrosine residues.